I welcome you to the session on basic virology. I am Dr. Richa Anjantiga. I am an assistant professor, Department of Microbiology from Chandulal Chandrakar Memorial Government Medical College, Kachandar, Durg. So the learning objectives in the session is morphology and nomenclature of viruses, the viral and host interaction, laboratory diagnosis of viral infection, and treatment and immunoprophylaxis of viral diseases. So, what are viruses? The characteristics that we look into are that they are obligate intracellular pathogens. These are those organisms which do not have any cellular organizations. So, the nucleic acid that they contain would be either DNA or RNA, but never both. These, since they lack the enzymes which are necessary for the protein and nucleic acid synthesis, they depend on the host cell machinery for those enzymes. The multiplication of the viruses is through a very complex process which is not as seen in bacteria via binary fission, and these viruses are organisms which are unaffected by antibacterial or antibiotics. As you can see in this table, it is differentiating between bacteria, mycoplasma, rickettsia and chlamydia with that of the viruses. As highlighted in red, viruses are those which are intracellular parasite, and other features that are also seen that they do not have an ATP generating metabolism, they do not contain ribosomes, and the sensitivity to antibiotics is not seen in them, and the sensitivity to interferons is visualized in the viruses. When we come to the morphology of viruses, the basic morphology that we see is virion, which is the extracellular infectious virus particle which contains nucleic acid, which can be single or double stranded, circular or linear, segmented or unsegmented. Now, based on the nucleic acid, it can be differentiated into DNA viruses or RNA viruses. Now, DNA viruses, they usually possess double-stranded DNA, which is signified by small d and s along with DNA in capitals. Now, in these DNA viruses, which does not have a double-stranded DNA, is parvovirus. Now, RNA viruses, they possess single-stranded RNA, except that in reovirus, in rotavirus, wherein they possess double-stranded, and in retroviruses, where they possess two copies of single-stranded RNA. The other essential part of the morphology is the capsid. Now, the capsid is a protein coat of the virus. This is made up of repeated protein subunits, polypeptides in nature, called capsomeres. The function of these capsid is to protect the nucleic acid core from the external environment. In non-enveloped viruses, we see the first initiation step in the viral replication through them, and these are pretty antigenic and very specific for each viruses. Now, the composition of capsid along with the virion is the combinations called nucleocapsid. When we see the symmetry in the morphology of viruses, it can be either icosahedral, helical or complex. In the icosahedral symmetry, you see the capsomeres which are arranged as if they lay on the faces of the icosahedral. As you can see in the image, 20 triangular facets with 12 corners or vertices is observed. It's a rigid structure, and these are seen in all DNA viruses except pox virus, and majority of the RNA viruses show this symmetry. In the helical symmetry, you see the capsomeres are coiled surrounding the nucleic acid, hence forming a spiral or a helix. This is a flexible structure, and you see this in RNA viruses like myxovirus, rhabdovirus, filovirus and bunyavirus. The complex symmetry is seen in pox virus, wherein you see neither of the above mentioned symmetry. Now, some viruses also have an envelope around it, the envelope that is containing of a lipoprotein cover, which is derived from the host cell membrane, from where the progeny virus is released through the process of budding. Now, the lipids, this is derived from the host cell membrane, whereas the proteins are coded by the virus. As you can see in the diagram, you can see certain structures colored in yellow and green. These are indicative of peplomers, which are projections on the surface of envelope. For example, in influenza virus, it has two peplomers, that is a hemagglutinin and a neuraminidase. Now, a mention of those viruses which are not enveloped. As mentioned, in DNA virus, you would have parvovirus, while in RNA virus, as mentioned, calicivirus, hepatitis A virus, to name a few. Now, when we differentiate the viruses based on the size, as you can see in the diagram, as compared to the red blood cell, which is about 8 nanometers in micrometers in size, you see the size of the current SARS-CoV-2. As these viruses are filterable through bacterial filters, they were first designated as smaller than that of the bacteria, and these cannot be visualized in a light compound microscope normally, but better visualized by an electron microscope, which helps in determining their size and shape. This diagram elicits the differential size between an Escherichia coli gram-negative bacteria with compared to that of the variable sizes of the virus, from the largest being the pox virus 300 nanometer in size and parvovirus 20 nanometer in size. And roughly the shape that we can see is usually spherical, but to name a few, we have some exceptionals like rabies virus, which is bullet shaped, and Ebola virus, which is filamentous in shape, and the rest of the shapes that can be visualized. When we differ the viroids and prions from viruses, the viroids are usually comprised of naked circular small single-stranded RNA, which does not have a capsid and is usually restricted to plants, and it depends on the host enzymes for replication. Prions, on the other end, is an abnormal infectious protein molecules which does not have a nucleic acid. Now, prions are highly resistant to physical and chemical agents, and they end up producing neurodegenerative condition in brains of humans called prion disease. Now, let's look into what the viruses are resistant of. With concerning the temperature, most of the viruses are heat labile, so we need to store it at -70 degrees centigrade. A better way of storing it with prolonged storage is through lyophilization. When we look into the pH changes and the nature of the viruses to it, enterovirus is one of the viruses which are resistant, but when we look into rhinoviruses, they are susceptible to it. So, a pH change can alter the existence of viruses. Radiation plays a role in inactivation through sunlight, UV rays and ionizing radiation. Disinfectants that we can use are as follows. When we look into the virucidal nature, phenols are weakly virucidal, while when we look into oxidizing agent, hydrogen peroxide, potassium permanganate, sodium hypochlorite, these are the disinfectants that we use in our day-to-day work. And chlorination of drinking water, which kills most of the viruses, but yes, it does get affected if there is organic matter present in the water. The presence of an envelope helps in seeing the resistance of the virus to a lipid solvent. An enveloped virus will be more sensitive to it, while a non-enveloped virus will be more resistant to a disinfectant. Now, coming to the nomenclature and classification, we will start with the DNA viruses. When we come to the nomenclature, when we look into the DNA viruses, if we look into the symmetry of it, we have an icosahedral structure in majority of them, with double-stranded DNA, which can be linear in majority, but as we look into the tabular form Hepadinaviridae family, we see a double-stranded circulate incomplete DNA type, which is usually seen in hepatitis B. And when we look into a different family, Papoviridae, this has double-stranded circular, and in this the envelope is absent and the symmetry stays to be icosahedral. A complex symmetry is seen in DNA viruses Poxviridae, wherein your DNA type is double-stranded and linear. So, variola and molluscum contagiosum virus will come under these family. Now, coming to RNA viruses. In RNA viruses, we can see that there is a differentiation of the RNA type in this, which will be either single-stranded positive sense. In this, you can see Piconaviridae, Calciviridae and Togaviridae family, wherein the envelope is present in Togaviridae, so it will be sensitive to lipid solvents. And when we look into the symmetry, it is icosahedral, and the representative viruses in each of this Piconaviridae, the most common you would know is poliovirus and enterovirus with hepatitis A virus, and Calciviridae you can see hepatitis E virus with Novak agent, and Togaviridae you can see rubella virus. The other RNA viruses that we can also look into, the table you can see the RNA type that is different, that is the negative sense single stranded RNA viruses, which is helical in shape. This is seen in Rhabdoviridae family. The most common virus that you encounter is rabies virus in this, and in Filoviridae family, in which you can see Marburg virus and Ebola virus. The next group of RNA viruses, this you can see that they all have helical symmetry with single stranded negative sense RNA strand. A special mention of Bunyaviridae, wherein you have 3 circular segments. In this you have the Hanta virus as an example. The next group of families, you can see that amongst these, Arenaviridae is a helical shaped single stranded negative sense RNA virus with 2 circular segments, while rest Retroviridae and Reoviridae, you can see the envelope is absent in the Reoviridae, which you see in rotavirus and reovirus. When we come into how do these viral particles multiply, so there are 6 essential steps that you have to look into, which is adsorption, penetration, uncoating, biosynthesis, maturation and release. The very first step, adsorption or attachment, it involves the receptor interaction. What do you have in this? Viruses, they have attachment sites on their envelopes or capsid protein, in which they bind to the complementary receptor site present on the host cell surface. For example, the HIV virus, on its surface you have GP glycoprotein 120, which binds to CD4 molecules on the host cells. Now, in influenza, another example is that the viral heme agglutinin peplomers that are present specifically on the virus, it binds to the glycoprotein receptors which is present on the surface of the respiratory epithelium. After adsorption, we have the penetration of the virus into the host cell, and this is through phagocytosis, membrane fusion or through injection of the nucleic acid. Phagocytosis and membrane fusion is what you see in majority of the viruses, and injection of the nucleic acid is what we observe in bacteriophages. Now, phagocytosis means it is a receptor mediated endocytosis of the virus inside the host cell, while in the membrane fusion, which is seen in HIV virus, this is fusion of the virus envelope with the envelope proteins into the plasma membrane of the host cell. After it has penetrated, there is uncoating. The uncoating step is through the action of the lysozymal enzymes of the host cells, the viral capsid gets separated and the nucleic acid is released into the cytoplasm. This is not seen in the bacteriophages. In biosynthesis, the viral components that are synthesized are as follows: nucleic acid, capsid protein, the enzymes which are required for the various stages of viral replication, and regulatory proteins to shut down the host cell metabolism. The site of the nucleic acid replication is which will lead and guide the biosynthesis of the components. In DNA viruses, this replication occurs in the nucleus, except that in pox virus, wherein the DNA virus occurs in the cytoplasm. And in case of RNA viruses, the replication occurs in cytoplasm, except that seen in the retroviruses and orthomyxoviruses, wherein it synthesizes RNA in the nucleus. In the biosynthesis of DNA viruses, as you can see, the first step is that of transcription of early genes, and thereafter from the DNA viruses, parental DNA, we get the early mRNAs from which through translation early proteins are synthesized. In the next step, which is replication of the viral DNA, you get multiple copies of parental DNA, and when the transcription of these parental DNA occurs, you get the late mRNAs. Thereafter, the translation leads to late protein production. Now, the fourth step is the assembly of the nucleocapsid, that is the composition of the proteins and the nucleic acid into daughter virions. Now, the biosynthesis of RNA viruses differ based on the genomic RNA, which can be either positive or negative sense, single or double stranded. In biosynthesis of RNA, it differs based on the positive or negative sense or based on the single stranded and double stranded. So, here we see type 1 positive sense single stranded RNA viruses, wherein through a single strand RNA, which is positive sense, the early viral proteins are made, which are then through translation, and through transcription with the action of RNA polymerase, we get negative single strand RNA. Thereafter, we get Through transcription, again a positive single strand RNA, and through translation you get late viral proteins. Now you have your viral proteins in it, and with your nucleic acid and a combination of daughter virions, and thereafter you get your positive single stranded RNA viruses. In type 2 negative sense, single stranded RNA viruses biosynthesis. First of all, through transcription you get a viral positive single stranded RNA through the help of viral RNA polymerase. The first viral proteins are produced through translation, which is based on the mRNA of the positive single strand RNA, and thereafter through transcription you get negative single stranded RNA. The composition of the nucleic acid and the viral proteins, you get the nucleocapsid for the daughter virions. In type 3 double stranded RNA, here through viral RNA polymerase, first of all the positive sense and the negative sense, both the RNAs are separated, and thereafter through the positive sense through translation you get the viral proteins. Thereafter through transcription you get multiple nucleic acid and the combination of the viral proteins and nucleocapsid in the daughter virions. Type 4 are retroviruses. Here we differ because you have a single stranded RNA which is positive, but in this you have reverse transcriptase enzyme which at different level plays a different role. From a single stranded RNA you get a double stranded RNA with the help of RNA dependent activity of reverse transcriptase enzyme. Thereafter the digestion between the RNA and the DNA strands occurs through the ribonuclease activity. We again get a single stranded DNA, and through DNA polymerase we get double stranded DNA. From there the double stranded DNA enters the host cell nucleus, and through viral integrase it incorporates itself into the parent host gene, and thereafter through transcription you get multiple mRNAs, and through host cell polymerase you get viral positive sense mRNAs too, and thereafter the daughter virions. In the fifth stage assembly, the viral nucleic acid and proteins are packaged together to form progeny viruses as nucleocapsids. This occurs in the host cell nucleus or cytoplasm. In DNA virus, you see the assembly happening in the nucleus, with the exception of herpesvirus and poxvirus in cytoplasm. The RNA viruses are assembled in the cytoplasm itself. In maturation, after the assembly, the maturation of the daughter virions take place either in the nucleus or cytoplasm or in the membranes, the Golgi or endoplasmic reticulum or the plasma membrane. Thereafter, in the last step is the release of the daughter virions, it occurs through the lysis of the host cells, which forms non-enveloped viruses or bacteriophages, or through budding from the host cell membranes, which leads to the formation of enveloped viruses. So there is a phenomena called eclipse phase. What is this eclipse phase? Is defined as an interval between the penetration of the virus into the host cell till the appearance of the first infectious virus progeny practical. Now during this period, you cannot see or demonstrate the virus inside the host cell. This takes about, the duration varies in bacteriophages to the animal viruses from minutes to hours. Now we come to viral multiplication. Now usually these are genetically defective viruses, so they cannot perform all the steps by themselves. So they need the help of the host machinery to help them to supplement in this. So now these defective viruses are like examples hepatitis D virus or adeno-associated satellite virus, which now adeno-associated satellite viruses require the help of adenoviruses, whereas hepatitis D virus require the help of hepatitis B virus. Over here, the von Magnus phenomena, which is basically an incomplete virus which is due to a defective assembly, and in this is only seen when the cells are infected with high dose of virus. So you see a high hemoglobin titer but low infectivity. And couple of other phenomena that you also see, non-permissive cells, which are virus infection in some cells which does not lead to that of production of infectious progeny. And another thing that you also see is abortive infection. In this you see the viral components are synthesized, but the assembly and the maturation is defective, hence you will not have a progeny, or there will be no release of such virions. So what are the few modifications you see after the multiplication? You'll see some modification in the genetic, which will be through two ways. That would be either through mutation or through recombination. So through mutation, you will see two types of mutants, which can be either conditioned lethal mutant or temperature sensitive mutants. Through recombination is usually when there are two different but related viruses and they together infect a single cell. So what is there in mutation? Now mutation, it usually occurs in each and every viral infection, which is usually 10 to the power minus 4 to 10 to the power minus 8 mutations per base per generation. It becomes only evident if it induces a readily observable property or it leads to a survival or death of the virus. Mutations occur spontaneously or it could be induced chemically or by physical agents. As I told before, types of mutations that you are going to see is conditional lethal mutant or temperature sensitive mutant. In conditional lethal mutant, it is certain specific conditions which are called as permissive conditions, which in those particular conditions the lethal mutant can grow, but it cannot grow in other conditions. Temperature sensitive mutant is the one that requires a specific temperature, a low permissive temperature, that of 28 to 31 degrees centigrade, but not a higher or restrictive temperature which is 37 degrees centigrade. An example of this is the TS mutant that is being used for the preparation of viral vaccines like the TS influenza vaccine. Now the interactions between virus genes is something that can be led genetically or non-genetically. This is when two or more virus particles infect the same host cells and a whole lot of interactions is seen as follows. In the genetic recombination, you see two different but related viruses of the same family which are simultaneously infecting a host cell. So there is an exchange of segments of nucleic acids between them and a hybrid or a recombinant virus is a resultant. So hybrids are something which possess a new gene but not found in both the parent viruses, and they are genetically stable and able to replicate. In recombination, you are able to see this in segmented RNA viruses like influenza rota or arenavirus. In this, the two strains of influenza virus, they infect a host cell. Gene exchange takes place between them and the RNA segments resulting in production of resortins. A property of viral interference is also seen as in the interaction of the viral and host. In this, the infection of a cell by one virus, it inhibits the subsequent infection by another virus or a simultaneous infection by another virus. This becomes an important mediator of interference through interferon. Now the interference does not occur with all the combinations, and many may infect and multiply together in a host cell. The most common clinical example is an oral polio vaccine. In this, the serotypes that are used, they interfere with the spread of the wild polio virus. This is essential as we use in the control of the polio outbreaks. Now enhancement is another property that we can also see, which is when mixed infection of cells is there, which can lead to either an increased virus yield or greater cytopathic effects. Now coming to virus host interaction in viral infections. In the transmission and spread of viruses, as we see ahead, it can be either through respiratory route or oral route, as seen in the organisms, which can be either affecting through local infection at the portal of entry or through the spread to a distant site. The other mode of transmission that you can also see is through cutaneous route, vector bites or animal bite. In vector bite and animal bite, we see straight away the spread to the distant site from the point of entry, which will be the source of bite. The other modes of transmission we also see is through sexual route or blood transfusion and via injection. Transplacental route and conjunctival routes are also seen as mode of transmission in the following viruses. So as you can see in this particular slide, the previous table had mentioned the portal of entry as a point. So the primary site of replication could be either the portal of entry or a site different from the portal of entry. So the ones that are producing infection at the local point, it could be either on the epithelial cells, wherein there is no viremia or spread of distant site. It will be having a shorter incubation period and shorter duration of immunity. but those in which the viruses they enter but spread the infection at a site distant from the portal of entry, this will be due to a spread via lymphatics or via blood or through neuronal spread. This is a diagram depicting pathogenesis of viral infection, wherein, as you can see, as soon as the infection occurs, if that infection is localized, it will be containing the infection at that particular site, and through the spread occurs via either the blood circulation or through lymphatics to distal site, where it can become an end, like you see in the brain, wherein the rabies virus, it has a human end as the last source. Now, a special word for the cell tropism of human viruses. This is basically the affinity of these viruses to specific cell type. Like you can see in the infection caused by Epstein-Barr viruses due to its tropism to the lymphocytes and infectious mononucleosis. In RNA viruses, you have mumps and measles specifically to their lymphocytes. While you can see for the neutrophils, you have influenza virus, and the tropism towards RBCs is through parvovirus B19 and cholera tick fever virus. Virus shedding is seen in certain viruses. Now, it could be either at the portal of entry, through blood, through an area nearby the target tissue organ, or no shedding at all. Now, at the portal of entry, due to local infection, like you can see in influenza virus, the virus could be shed through respiratory secretions. Now, through blood, it can be either through a vector bite or through transfusion or needle pricks that you can observe in hepatitis B virus or arboviruses. Virus shedding near the target tissue is seen in that examples of varicella zoster, mumps or cytomegalovirus, which is nearby the skin, salivary gland or kidney. Now, the manifestations of the viral infection. It all depends on the incubation period, from the time of entry of virus into the body and appearance of the first clinical manifestation. So, depending on the distance between the two, it could be either shorter, that means through the site of entry you see that particular lesion, or longer incubation period, that means the target organ is away, like you see in rabies virus. The bite is in one place and the affection is elsewhere. In this, you have an exception of dengue virus and hepatitis B virus and the other factors that also play, and they depend on the incubation virus is through host immune response and nature of the virus. So, the clinical manifestation could be subclinical, which is inapparent, or could be an apparent one, dividing it into an acute, subacute or chronic nature depending on the onset of illness. Now, the clinical manifestation, depending on the body sites, you will be able to see the example of the viruses as well as the target body sites. So, what happens between the virus and the host at cellular level? Either there will be no apparent cellular damage, or there can be cell death due to cytocidal as seen in poliovirus, cell lysis, cell proliferation as seen in molluscum contagiosum, malignant transformation, and also cytopathic effect that we see in vitro when we grow it in tissue culture. Now, a factor of failed infection can also take place. What happens in this is that there is infection of the host cells, but this is permissive. That means you do not have surface receptors or the machineries to support viral replication, hence it becomes a failed infection. So, what happens in cell death is through the inhibition of host cell DNA, like that seen in herpes virus, or inhibition of host cell protein synthesis as in poliovirus, you will be able to see different mechanisms of cell death. Fusion, syncytia formation and immune mediated lysis is also seen as other mechanisms. Infection without cell death is also seen in two different ways. It can be either a steady state infection in which you see the virus and host cells replicating independently and without any cellular injury. Now, the persistent viral infection is as that seen in latent infection caused by herpes virus with periodic exacerbation, or that latency seen in HIV infection. Now, you can also see latency in slow virus infection as well as essentially persistent tolerant infection seen in lymphocytic choriomeningitis virus, which is seen affecting mice. Now, the morphological changes in the host cells. Now, in the virus host interaction, there are certain morphological changes in the host cells. These are evident by inclusion bodies, which are generally aggregates of virions of viral proteins which are formed by viral replication, and we can see them through the altered staining property. These are very specific for certain viral infection, and they are distinct structures that we can see in vitro. So how do we go about seeing them? Through light compound microscope, in which you can see them either intracytoplasmic as you see in pox virus, intranuclear as you see in herpes virus, and cytoplasmic as well as nuclear as you see in measles virus. These can be either acidophilic, pink as seen in Giemsa stain, as in adenovirus, which can be basophilic. Now, as per the location, it can be either intracytoplasmic, intranuclear. In intranuclear, you have your Cowdery type A inclusion bodies which are variable and granular, Cowdery type B inclusion bodies which are circumscribed and multiple, or it could be both intracytoplasmic or intranuclear also. Here are few examples of the same. This is a picture depicting the Lipschitz bodies seen in herpes virus. So, the host cell response seen in viral infection is usually affected by the virulence of the infected strain and the host resistance. When you see the immunological response, due to the good antigenic nature of the virions, it is inducing both humoral as well as cellular immunity. The immunity in viral infection can be elicited through virus neutralization caused by the antibodies, which will lead to prevention of absorption of the virus to the cell receptors. Now, this will cause the degeneration of the virus, and it also prevents the release of the progeny virus from the infected cells. Now, the complements which acts on the antibodies causes surface damage to enveloped virions and producing cytolysis of those cells which are infected by virus. The cell mediated immunity plays an essential role along with immunity to reinfection. Now, certain infection can also cause suppression of immunity. Other factors which we also see is phagocytosis and fever acting as natural defense, corticosteroids enhancing those infections. Like in measles, even the role of malnutrition plays. Now coming to interferon. So, host-coded proteins which are produced by cells due to induction by viral or non-viral inducers. So, when there is an exposure to interferons, these cells they produce translation inhibiting the protein, which causes selective inhibition of the viral mRNA synthesis. So, what is this? This is specific to species, not virus specific, and these are caused by three types of interferon, which are alpha, beta, gamma, and the biological effects of this is leading to antiviral or immunoregulatory modules. Thank you.